As this lecture begins, there is no need to focus sharply or hold on to every detail. The subject we are approaching unfolds at a scale far smaller than everyday life, and it invites a slower, softer kind of attention. Microbiology, and the science of antibiotics within it, concerns forms of life so small they pass unnoticed beneath our senses. Yet they have always been present, quietly shaping the conditions of health, illness, and balance. To understand them does not require urgency or effort, only a willingness to drift gently into a world that has always existed alongside our own. Long before microscopes revealed their presence, microscopic organisms lived in the soil beneath our feet, in the water that flows through rivers and oceans, and within the bodies of plants, animals, and humans. They are ancient companions, older than forests, older than animals, older even than complex cells. For most of human history, these tiny forms of life were unknown, not because they were absent, but because they were beyond perception. And yet, despite their invisibility, they quietly participated in cycles of decay and renewal, in digestion, in fermentation, and in the slow shaping of the living world. When we speak of antibiotics, we are speaking about a human effort to understand and gently influence this microscopic realm. Antibiotics are not abstract inventions imposed upon nature, but discoveries drawn from it. They are substances that interact with bacteria, one particular group of microscopic life, in ways that can restore balance when certain bacterial populations grow where they should not. This is not a story of conquest or battle, but of observation, learning, and careful adjustment. At the beginning of this journey, it helps to soften the idea of scale. The microscopic world is not simply a smaller version of our own. It operates according to rhythms and patterns that feel almost timeless. A single grain of soil may contain more bacteria than there are humans on the planet. A single drop of water may hold countless living cells, drifting slowly, responding to light, temperature, and chemical signals. These organisms do not rush. They grow, divide, and interact in ways governed by chemistry and environment, unfolding steadily over time. Within the human body, this invisible world is always present. From the moment of birth, bacteria begin to settle on the skin, in the mouth, and throughout the digestive system. Most of these organisms are not harmful. In fact, many perform quiet tasks that support digestion, help regulate the immune system, and occupy spaces that might otherwise be taken by less helpful microbes. The body, in this sense, is not a solitary organism, but a shared environment. The need for antibiotics arises only when this balance is disturbed. Certain bacteria, under certain conditions, can multiply in ways that strain the body's systems. When this happens, antibiotics may be introduced to gently slow or stop the growth of those specific bacteria, giving the body space to recover. This process, when understood calmly, is less about elimination and more about restoration. It is an intervention meant to return a system to its natural state of equilibrium. To appreciate antibiotics fully, it is useful to let go of modern associations with urgency or crisis. Instead, imagine a slow, methodical science rooted in patience. The earliest antibiotics were not synthesized in laboratories, but discovered in nature, often by accident. Soil-dwelling bacteria and fungi have been producing antibacterial substances for millions of years, using them not as weapons, but as tools to manage their immediate surroundings. Humans, through careful observation, eventually learned to recognize and refine these substances for medical use. This learning process mirrors much of scientific discovery. It begins not with control, but with curiosity. Scientists noticed that certain molds prevented bacteria from growing nearby, or that certain soil samples inhibited infection. Over time, these observations accumulated into knowledge, and knowledge gradually became practice. Antibiotics emerged as an extension of natural processes, guided by human understanding rather than imposed upon the living world. As we move deeper into this topic in later parts, there will be explanations of how bacteria are structured, how they grow, and how antibiotics interact with their cellular machinery. But for now, it is enough to rest in the broader picture. The microscopic world is vast, diverse, and mostly peaceful. Antibiotics represent a careful conversation with that world, one that requires respect, restraint, and an appreciation for complexity. It is also worth noting that microbiology does not isolate bacteria as villains or heroes. It simply observes them as living systems responding to their environments. Bacteria adapt, reproduce, and change over generations, just as all living organisms do. Antibiotics, when introduced, become part of that environment, influencing which bacteria thrive and which slow down. This dynamic interaction is neither static nor final. It is ongoing and gradual. In this sense, the science of antibiotics is not fixed knowledge, but a living field that continues to evolve. New discoveries build upon older ones, and understanding deepens as tools improve. Yet the fundamental principles remain calm and consistent. Life at the microscopic scale responds to chemistry, structure, and time. Antibiotics work not through force, but through specificity, interacting with features unique to bacterial cells. As you listen, there is no need to visualize diagrams or remember terminology. Instead, imagine this knowledge as a quiet landscape, unfolding slowly. Picture soil rich with unseen life, water carrying drifting cells, and the human body as a warm, stable environment shaped by countless microscopic interactions. Antibiotics move gently within this picture, not as disruptors, but as carefully chosen influences. This first part serves simply as an invitation to set aside the visible world for a while and to acknowledge the steady presence of life beyond sight. In doing so, the science becomes less technical and more intuitive, less about memorization and more about understanding relationships. The microscopic world does not demand attention. It rewards quiet awareness. As this introduction settles, the mind can begin to relax into the subject. The coming parts will explore bacteria themselves, the nature of infection, and the way antibiotics function, all with the same measured pace. For now, it is enough to recognize that beneath the surface of everyday life exists a vast, ancient, and remarkably orderly world, one that has always been there, patiently waiting to be understood, to life too small to see. As attention drifts further into the microscopic world, it becomes easier to release the habits of thinking shaped by what is large, visible, and immediate. Life at this scale does not announce itself. It moves quietly, continuously, and without spectacle. Microorganisms exist everywhere, not clustered in distant or unusual places, but woven into the most ordinary surroundings. They inhabit the air that passes gently through a room, the thin film of moisture on a leaf, the grains of dust resting on a windowsill, and the warm interior spaces of living bodies. To say that microorganisms are everywhere is not an exaggeration, but a simple description of reality. Soil, for example, is not merely a mixture of minerals and organic debris. It is alive with bacteria, fungi, and other microscopic forms. These organisms break down fallen leaves and dead roots, releasing nutrients back into the environment. Without this invisible labor, plant life would struggle to renew itself, and ecosystems would gradually stall. What appears still and silent at the surface is, at a smaller scale, quietly active. Water tells a similar story. Oceans, lakes, rivers, and even rain droplets contain countless microscopic organisms drifting with currents and responding to subtle changes in temperature, light, and chemistry. Some float freely, while others attach themselves to surfaces, forming thin layers of living material. In the vastness of the ocean, microscopic life forms the foundation of entire food webs, supporting creatures many orders of magnitude larger. These tiny organisms do not hurry, yet over time they shape the chemistry of the planet itself. Air, too, carries life too small to see. Bacteria and fungal spores rise from soil and water, traveling on gentle air currents across continents and oceans. They settle slowly, often unnoticed, becoming part of new environments. This movement is not purposeful in the human sense, but it is persistent. Microorganisms disperse gradually, responding to forces like wind and humidity. Within the human body, this invisible presence becomes even more intimate. From the skin to the digestive tract, microorganisms live in quiet association with human cells. The skin alone supports a diverse collection of bacteria adapted to its dryness, temperature, and chemical makeup. These organisms occupy space, consume available resources, and in doing so, often prevent less compatible microbes from gaining a foothold. Their presence is steady and largely beneficial, though it passes entirely below conscious awareness. Inside the digestive system, the number of microorganisms increases dramatically. The intestines, in particular, provide a warm, nutrient-rich environment where bacteria thrive. These bacteria assist in breaking down complex molecules, producing vitamins, and interacting with immune cells that line the gut. Over time, this relationship becomes finely tuned. The body provides a stable environment, and in return, these microbes contribute to normal physiological function. It is a quiet partnership maintained through balance rather than control. What is striking about this microscopic world is not only its abundance, but its diversity. Bacteria alone exist in countless shapes, sizes, and metabolic styles. Some prefer oxygen-rich environments, while others thrive only where oxygen is absent. Some feed on sugars, others on minerals, and still others on compounds that would be toxic to larger organisms. This diversity allows microorganisms to occupy nearly every ecological niche imaginable, from deep-sea vents to frozen tundra. Despite this diversity, microorganisms share a common simplicity. Their lives unfold at the cellular level, governed by chemical reactions and physical constraints. They do not plan or anticipate. They respond. When conditions are favorable, they grow and divide. When conditions change, growth slows, or cells enter dormant states, waiting patiently for improvement. This responsiveness, rather than complexity of thought, defines their existence. It is within this responsive world that antibiotics eventually find their place. But before antibiotics are introduced, it is important to understand that most microorganisms coexist without causing harm. Illness arises not from their mere presence, but from changes in context. A normally balanced population may grow too large if conditions shift. These are not acts of intention, but consequences of altered environments. The human immune system plays a constant, gentle role in maintaining this balance. Immune cells monitor tissues, responding to signs of disruption rather than to the simple presence of microbes. Much of the time, this monitoring proceeds quietly, without noticeable symptoms. Even then, the response is gradual, involving communication between cells rather than sudden action. Microbiology, when viewed calmly, reveals a world defined by gradual change. Microorganisms evolve slowly over generations, adapting to their environments through small adjustments. They exchange genetic material, acquire new traits, and lose others, all over spans of time that are imperceptible to daily experience. Antibiotics enter this picture as one more environmental factor, influencing which organisms flourish and which recede. It is helpful here to let go of any notion that the microscopic world is chaotic or hostile. In reality, it is remarkably ordered. Chemical gradients guide movement. Temperature, moisture, and available nutrients define boundaries. Even competition among microorganisms unfolds quietly through subtle chemical signals and resource use rather than overt confrontation. The overall effect is a dynamic equilibrium, constantly adjusting yet rarely dramatic. Human awareness of this world arrived relatively late. Only with the development of lenses capable of magnifying the very small did these organisms become visible. When they did, it transformed understanding, not by revealing danger everywhere. The realization that life existed beyond sight did not diminish the visible world. It enriched it. As this awareness grew, so did the ability to interact with microorganisms intentionally. Antibiotics would eventually become one such interaction, but they rest upon this deeper understanding of ubiquity and balance. To influence microscopic life responsibly requires an appreciation for its constant presence and its quiet role in sustaining larger systems. For now, it is enough to remain with the idea that life too small to see is not separate from daily existence. It is embedded within it, shaping and supporting it in subtle ways. Every breath, every meal, every moment of contact with the environment involves countless unseen interactions. Most pass without notice, and most proceed smoothly. As this part comes to a close, the mind can rest in the sense of continuity that defines the microscopic world. Nothing begins abruptly, and nothing ends suddenly. Change happens slowly, through accumulation and adjustment. In the next part, attention will narrow gently to bacteria themselves, examining their structure and simplicity. Until then, the invisible world can remain what it has always been: present, patient, and quietly alive. 3. Bacteria as simple living cells. As attention settles more deeply into the microscopic world, it becomes natural to focus on one particular form of life that plays a central role in the story of antibiotics: bacteria. Bacteria are among the simplest living organisms on Earth, yet they are also among the most enduring. They appeared billions of years ago, long before plants and animals, and they have persisted through dramatic changes in the planet's atmosphere, climate, and geography. Their simplicity is not a weakness, but a quiet strength that has allowed them to adapt and survive across immense spans of time. A bacterium is a single living cell. Unlike the cells that make up plants and animals, bacterial cells do not contain a nucleus or complex internal compartments. Everything they need to live is contained within a small, efficient structure bounded by a cell membrane and, in most cases, a protective cell wall. This simplicity allows bacteria to function with minimal resources, responding directly to their surroundings without layers of internal regulation. Within this small cellular space, life unfolds through chemistry. The bacterial cell contains genetic material, usually in the form of a single circular strand of DNA, floating freely within the cell rather than enclosed within a nucleus. This DNA carries the instructions needed for growth, maintenance, and reproduction. There is no separation between storage and use of information. When conditions are right, genes are expressed directly, guiding the production of proteins that keep the cell functioning. Proteins do most of the work inside a bacterium. They act as enzymes that speed up chemical reactions, structural components that maintain shape, and transporters that move substances in and out of the cell. These proteins are assembled by tiny molecular structures called ribosomes, which are smaller and simpler than those found in human cells. This difference, subtle as it may seem, becomes important later when considering how antibiotics can affect bacteria without harming human tissue. The bacterial cell wall is another defining feature. It provides structure and protection, helping the cell maintain its shape in changing environments. This wall is made of a sturdy material not found in human cells, giving bacteria both resilience and vulnerability. The presence of this unique structure allows antibiotics to target bacteria specifically. Despite their simplicity, bacteria are remarkably diverse. Some are shaped like tiny spheres, others like rods, spirals, or gentle curves. These shapes are not decorative. They influence how bacteria move, attach to surfaces, and interact with their environments. Some bacteria possess small, whip-like structures called flagella that allow them to move slowly through liquid, while others remain still, relying on currents to carry them. Bacteria do not require complex sensory organs to respond to their surroundings. When nutrients are abundant, growth proceeds. When resources become scarce, activity slows. This responsiveness allows bacteria to occupy environments that are constantly changing, from the human gut to soil warmed by the sun and cooled by night. Reproduction in bacteria is equally straightforward. Most bacteria reproduce through a process called binary fission, in which a single cell grows, duplicates its genetic material, and divides into two identical cells. This process unfolds steadily, without ceremony or urgency. Under favorable conditions, it can happen relatively quickly, but it is always governed by the availability of nutrients and suitable environmental conditions. When circumstances are less favorable, division slows or stops altogether. This ability to pause is another quiet strength. Many bacteria can enter dormant states, reducing metabolic activity until conditions improve. In this state, they may persist for long periods, neither growing nor dying, simply waiting. This patience allows bacterial populations to survive drought, cold, and scarcity, resuming activity only when the environment becomes supportive again. It is important to remember that most bacteria are not harmful. Many play essential roles in ecosystems, breaking down organic matter, recycling nutrients, and supporting the growth of plants. In the human body, beneficial bacteria contribute to digestion and immune regulation, forming stable communities that change slowly over time. These bacteria are not invaders. They are residents, shaped by and adapted to their environments. Illness associated with bacteria arises not from their basic nature, but from context. A bacterium well suited to one environment may cause problems if it ends up somewhere else. Similarly, a normally harmless population may grow excessively if balance is disrupted. These outcomes are not acts of intention, but natural consequences of altered conditions. From the perspective of antibiotics, the simplicity of bacterial cells is key. Because bacteria rely on structures and processes that differ from those of human cells, it becomes possible to influence bacterial growth selectively. Antibiotics are designed to interact with features that bacteria depend on, such as their cell walls or protein-making machinery. This selectivity is not absolute, and it requires careful use, but it rests upon fundamental biological differences. Understanding bacteria as simple living cells helps clarify why antibiotics work the way they do and why they must be used thoughtfully. Bacteria are not complex organisms with multiple backup systems. When a critical process is disrupted, growth slows or stops, and the population changes accordingly. At the same time, bacterial simplicity does not mean fragility. Small changes in genetic material can accumulate, leading to variations that respond differently to environmental pressures, including the presence of antibiotics. This adaptive capacity will become more relevant later, but for now it can be acknowledged as part of the quiet resilience that defines bacterial life. As this understanding settles, it becomes easier to view bacteria without fear or fascination. They are simply living cells, following the same basic principles that govern all life. They take in energy, maintain internal conditions, respond to their environment, and reproduce when able. Their scale is smaller, their structure simpler, but their existence is no less orderly. In the calm context of microbiology, bacteria appear not as threats, but as participants in a larger system. Antibiotics enter this system as one influence among many, altering conditions in specific ways. To appreciate this interaction fully, It helps first to understand the nature of the bacterial cell itself, its simplicity, its adaptability, and its place within the broader tapestry of life. As this part comes to a close, the image of bacteria as small, self-contained units of life can remain gently in mind. In the next part, attention will turn to how these cells grow and multiply over time, exploring the rhythms of bacterial reproduction with the same unhurried pace. For now, the simple structure of bacterial life offers a quiet foundation upon which the rest of the story can unfold. 4. How bacteria grow and multiply. With an understanding of bacteria as simple living cells, it becomes natural to turn attention toward the way they grow and multiply. At this microscopic scale, growth does not involve visible movement or dramatic change. Instead, it unfolds quietly, governed by chemistry, energy availability, and time. Bacterial growth is steady and methodical, responding gently to conditions in the surrounding environment. For a single bacterial cell, growth begins with the accumulation of resources. Nutrients pass through the cell membrane, guided by proteins that recognize and transport specific molecules. Inside the cell, these nutrients are processed, converted into energy, and used to build new cellular components. Proteins are assembled, membranes expand, and internal structures duplicate. Nothing happens all at once. Each step proceeds only as quickly as the available energy allows. When conditions are favorable, growth leads naturally to reproduction. Most bacteria reproduce through a process known as binary fission, a term that simply describes the division of one cell into two. The process begins with the duplication of the bacterial chromosome. This circular strand of DNA is copied carefully, ensuring that each new cell will receive a complete set of genetic instructions. As the DNA duplicates, the cell elongates slightly. Gradually, the cell membrane begins to pinch inward at the center. New cell wall material is synthesized and laid down, forming a barrier between the two emerging cells. Eventually, the division completes, and two independent bacterial cells exist where there was once one. Each new cell is, under ideal circumstances, nearly identical to the original, carrying the same genetic information and the same basic structure. This process can occur relatively quickly, but it is never rushed. The speed of bacterial division depends entirely on environmental conditions. Temperature, nutrient availability, moisture, and chemical surroundings all influence how rapidly growth proceeds. In warm, nutrient-rich environments, some bacteria may divide every 20 minutes or so. In less favorable conditions, division may take hours, days, or may pause altogether. It is important to emphasize that bacterial reproduction is not constant or uncontrolled. Bacteria do not divide endlessly without regard for their surroundings. As populations grow, resources become limited. Waste products accumulate. Space becomes crowded. These changes naturally slow growth, leading populations toward a stable balance with their environment. In this way, bacterial growth follows the same ecological principles that govern larger organisms, simply compressed into a smaller scale. When conditions deteriorate significantly, many bacteria shift into survival mode. Metabolic activity slows, and reproduction stops. Some species can form specialized structures that protect their genetic material, allowing them to endure extreme heat, dryness, or lack of nutrients. In these states, bacteria may persist quietly for long periods, neither growing nor dying, simply waiting for conditions to improve. This ability to pause is essential to understanding bacterial resilience. Growth is not a constant drive, but a response to opportunity. When opportunity fades, activity recedes. This pattern contributes to the stability of microbial populations over time, allowing them to endure environmental fluctuations without dramatic collapse. Within the human body, bacterial growth is shaped by similar factors. Temperature remains relatively stable, and nutrients are supplied regularly, creating conditions that support consistent microbial activity. However, the immune system plays an ongoing role in regulating growth. Immune cells monitor bacterial populations, responding when growth exceeds what the environment can support comfortably. This regulation usually proceeds quietly, without noticeable symptoms. When bacterial growth becomes excessive or occurs in inappropriate locations, the body responds more actively. Inflammation, fever, and other symptoms reflect efforts to restore balance rather than eliminate bacteria entirely. Antibiotics, when introduced, become an additional influence on growth, slowing or stopping reproduction in targeted populations. From the perspective of antibiotics, bacterial growth represents a critical point of intervention. Antibiotics do not usually kill bacteria instantly. Instead, many interfere with the processes required for growth and division. By disrupting cell wall construction or protein synthesis, antibiotics prevent bacteria from reproducing effectively. Over time, this slows population expansion, allowing natural regulatory mechanisms to regain control. This gradual effect aligns with the natural rhythms of bacterial life. Growth and division are continuous, but measured processes. Interrupting them does not cause sudden collapse, but rather a gradual shift in population dynamics. This is one reason antibiotic treatment typically spans several days rather than acting immediately. It is also during growth and division that variation can arise. Although binary fission produces nearly identical cells, small differences occasionally occur. Errors in DNA replication, though rare, can introduce changes. Over many generations, these small variations accumulate, contributing to diversity within bacterial populations. Most changes have little effect, but some may alter how bacteria respond to environmental pressures, including antibiotics. This capacity for variation is not purposeful or strategic. It is simply a byproduct of reproduction over time. Bacteria do not anticipate challenges. They respond to them as they arise. When conditions change, populations shift gradually, favoring cells better suited to the new environment. This process unfolds quietly, generation after generation. Understanding bacterial growth in this calm, ecological context helps dissolve the idea of unchecked expansion. Bacteria grow when conditions support growth and slow when they do not. Their populations rise and fall gently, influenced by resource availability, environmental signals, and interactions with other organisms. In nature, bacterial growth supports broader systems. In soil, growth contributes to nutrient cycling. In water, it underpins food webs. In the human body, it supports digestion and immune balance. Only when growth becomes misaligned with context does it contribute to illness, and even then, the response is typically measured rather than extreme. As this part settles, the image of bacterial growth can remain simple and steady. A single cell absorbs nutrients, expands, duplicates its genetic material, and divides, producing two cells that continue the process. This cycle repeats as long as conditions allow, slowing or stopping when they do not. There is no urgency in this rhythm, only responsiveness. In the next part, attention will turn to moments when this balance shifts more noticeably, exploring how bacterial growth can contribute to illness under certain circumstances. For now, the quiet regularity of bacterial multiplication offers a calm foundation for understanding how antibiotics interact with living systems, not as abrupt forces, but as influences that gently alter the conditions under which growth occurs. 5. When balance shifts toward illness. In the quiet rhythm of the microscopic world, balance is the natural state. Bacteria grow, slow, and persist in ways that generally align with the environments they inhabit. Within the human body, this balance is usually maintained without conscious awareness. Countless bacterial cells live and function alongside human cells, neither harming nor helping in dramatic ways. Illness begins not as a sudden event, but as a gradual shift in this equilibrium, when conditions change in ways that favor certain bacteria more than the body can comfortably accommodate. These shifts can arise from many sources, often subtle and unremarkable on their own. Or a temporary weakening of immune defenses may alter the local environment just enough to allow a bacterial population to grow where it normally would not. The bacteria themselves have not changed in character. They are responding to opportunity, expanding into available space as they are naturally inclined to do. Within tissues that are normally free of large numbers of bacteria, even a small increase can trigger a response. The immune system, finely attuned to chemical signals, detects this change and begins to adjust. White blood cells migrate toward the affected area. Blood flow increases slightly. Chemical messengers are released. These changes are not acts of aggression, but efforts to restore the familiar balance that the body prefers. The sensations associated with illness often arise from this response rather than from the bacteria themselves. Warmth, redness, swelling, and discomfort reflect increased circulation and cellular activity. Fever, when it occurs, gently alters body temperature in ways that can slow bacterial growth. Fatigue encourages rest, conserving energy for immune processes. These experiences, though sometimes unpleasant, are part of a coordinated attempt to return the system to stability. Most of the time, these adjustments are sufficient. The immune system resolves the imbalance, bacterial populations recede, and normal conditions resume. This process unfolds quietly, often without external intervention. In many cases, the body never reaches a point where assistance is required, and the episode passes as a brief disruption rather than a lasting problem. However, there are circumstances in which the shift toward imbalance exceeds the body's capacity to respond efficiently. This may occur when bacterial growth is particularly rapid, when bacteria reach sensitive tissues, or when the immune system is compromised or distracted. Age, underlying health conditions, and environmental factors all influence how readily balance can be restored. In such cases, bacterial populations may continue to expand, not out of intent, but because conditions allow it. Nutrients remain available. Immune responses may be delayed or insufficient. The local environment becomes increasingly supportive of growth rather than restraint. As this unfolds, symptoms may intensify, reflecting the ongoing effort of the body to correct the imbalance. It is at this point that antibiotics may enter the picture. Their role is not to replace the immune system, but to assist it by altering the conditions under which bacteria grow. By slowing reproduction or interfering with essential bacterial processes, antibiotics reduce the pressure on the body's regulatory systems. This creates space for immune responses to regain effectiveness and for balance to be reestablished. Understanding illness in this way shifts the perspective from conflict to adjustment. Bacteria are not invading forces with intentions or strategies. They are cells responding to changes in their environment. When those changes occur within the human body, the result may be discomfort or dysfunction, but the underlying process remains one of ecological imbalance rather than confrontation. This view also helps explain why the same bacteria can exist harmlessly in one context and contribute to illness in another. A bacterium that lives quietly on the skin may cause infection if it enters deeper tissues. A population that is stable in the gut may become problematic if it grows unchecked or migrates to areas where it does not belong. The difference lies not in the organism itself, but in the surrounding conditions. The body's internal environments are normally well defined. Barriers such as skin, mucous membranes, and tightly joined cells maintain separation between spaces. Fluids flow in controlled ways. Chemical signals guide immune surveillance. When these systems function smoothly, balance is maintained. When barriers are disrupted or signals altered, opportunities for imbalance arise. Antibiotics, when used, become one more environmental factor influencing this delicate system. They do not discriminate between helpful and unhelpful bacteria perfectly. Instead, they act broadly on certain bacterial features, reducing populations in ways that can tip conditions back towards stability. This influence is temporary and context-dependent, working best when combined with the body's own regulatory efforts. The gradual nature of illness is important to hold in mind. Rarely does balance collapse instantly. More often, it shifts incrementally, with early signs appearing before more noticeable symptoms develop. This gradual progression mirrors the slow rhythms of bacterial growth itself. Both illness and recovery unfold over time, shaped by many small interactions rather than a single defining moment. From this perspective, the goal of treatment is not eradication, but moderation. Complete elimination of bacteria is neither possible nor desirable. Life at the microscopic scale is too deeply integrated into human biology for that. Instead, the aim is to restore proportions, to reduce excessive growth, and allow cooperative relationships to resume. This understanding also underscores the importance of restraint. Using antibiotics when they are not needed introduces unnecessary changes into microbial communities, altering balance where none was truly lost. Over time, such disruptions can make future imbalances more difficult to correct. Thoughtful use respects the underlying complexity of the system rather than attempting to override it. As this part draws to a close, it may help to picture illness not as an external force acting upon the body, but as an internal shift, a temporary misalignment within a living system that is usually stable. Bacteria respond to opportunity. The body responds to change. Antibiotics, when appropriate, assist by gently nudging conditions back toward equilibrium. In the next part, attention will turn to how humans first encountered bacterial illness historically, long before antibiotics existed, and how understanding grew slowly through observation. For now, the idea of illness as imbalance can settle quietly, offering a calm framework for understanding why antibiotics exist and how they fit into the broader rhythm of living systems. 6. Early human encounters with infection. Long before bacteria were known to exist, long before microscopes revealed the hidden architecture of cells, humans were already living alongside microbial life and responding to its effects. Illness was a familiar part of existence, yet its causes remained invisible and mysterious. People observed patterns, noticed symptoms, and developed explanations rooted in the knowledge available to them at the time. These early encounters with infection were not guided by certainty, but by careful attention to experience and gradual learning passed from one generation to the next. In early human societies, illness was often understood through the lens of environment and balance. Changes in weather, water quality, food availability. Communities noticed that certain seasons brought more sickness, that wounds sometimes healed quietly while others worsened, and that crowded living conditions often coincided with more frequent illness. These observations were accurate, even if the underlying mechanisms remained unknown. Without knowledge of bacteria, explanations for infection were necessarily indirect. Some cultures attributed illness to imbalances within the body, others to external influences such as spoiled air, contaminated water, or unseen forces carried by the environment. These ideas, though incomplete, reflected genuine attempts to make sense of recurring patterns. They emphasized prevention through cleanliness, rest, and moderation, practices that often reduced the likelihood of infection, even without understanding why. Wounds offered some of the clearest early encounters with bacterial illness. A cut or scrape might heal uneventfully, or it might become red, swollen, and painful. Over time, people learned that keeping wounds clean and protected improved outcomes. Honey, plant resins, and certain oils were applied not because bacteria were known, but because these substances seemed to help. In retrospect, many of these treatments had mild antibacterial properties. offering early, intuitive forms of infection control. Food preservation provided another window into microbial activity. Fermentation, drying, and salting were developed through observation and experimentation. People noticed that certain methods prevented spoilage and illness, while others did not. These practices worked by shaping microbial growth, encouraging some organisms while inhibiting others. Again, the effects were understood through results rather than mechanisms. Water sources were also carefully observed. Communities learned that water from certain streams or wells was safer than water from stagnant pools. Boiling water, though not universally practiced, was recognized in some cultures as a way to improve safety. These habits reduced exposure to harmful bacteria without naming them, reflecting an early awareness of environmental influence on health. As societies became more complex, written records began to capture medical thought. Ancient texts describe symptoms that align closely with bacterial infections, even if they are framed in symbolic or philosophical terms. Fever, inflammation, and localized pain were recognized as meaningful signs. Treatments focused on supporting the body through rest, nourishment, and gentle interventions, rather than aggressive measures. The idea of balance remained central. Health was seen as a state of harmony within the body and between the body and its surroundings. Illness represented a disruption of this harmony. While the causes were interpreted differently across cultures, the emphasis on restoring balance rather than eliminating an enemy shaped early medical approaches. Hospitals and organized care, where they existed, brought both help and challenge. Concentrating ill individuals in one place allowed for focused attention, but also created conditions where infections could spread more easily. Without understanding microbial transmission, these environments sometimes amplified illness rather than containing it. Over time, patterns emerged, and practices slowly adapted. Cleanliness became an important theme, though its implementation varied. Bathing, laundering, and ventilation were encouraged in many traditions, not out of fear of bacteria, but from an association between cleanliness and well-being. These measures, though imperfect, often reduced infection risk by limiting microbial growth and transmission. Surgical practices offer a particularly striking example of early encounters with infection. Procedures were performed with skill and care, yet post-operative infections were common and often severe. Surgeons recognized that outcomes varied, but without knowledge of microbes, the reasons remained elusive. Some noticed that instruments and surroundings mattered, leading to gradual improvements in technique even before formal germ theory emerged. Throughout these centuries, the relationship between humans and infection remained largely observational. People responded to what they could see and feel, adjusting behaviors based on outcomes. This slow accumulation of practical knowledge laid the groundwork for later scientific advances. When bacteria were finally identified, they did not overturn all prior understanding, but rather explained patterns that had long been noticed. The transition from mystery to mechanism did not happen suddenly. It unfolded as new tools allowed humans to see what had always been there. Early microscopes revealed tiny moving forms in water and bodily fluids, sparking curiosity rather than immediate clarity. These observations suggested that life existed beyond sight. But linking these organisms directly to illness took time and careful experimentation. Once connections began to form, earlier practices took on new meaning. Cleanliness, wound care, and food preservation could now be understood in terms of microbial growth and control. What had once been tradition or intuition became supported by emerging biological knowledge. This shift did not erase the past. Antibiotics, when they eventually appeared, represented a continuation of this long process rather than a sudden departure. They offered a more precise way to influence bacterial growth, but they rested on centuries of observation about illness, recovery, and balance. The calm, methodical nature of this progression reflects the broader rhythm of medical understanding, advancing through patience rather than haste. As this part comes to rest, it may help to imagine early humans encountering infection not with fear, but with attentive curiosity. They noticed patterns, adjusted behaviors, and shared knowledge within their communities. Their experiences form the quiet background against which modern microbiology developed. In the next part, attention will turn toward the natural world itself, exploring how antibacterial substances existed long before humans identified or refined them. For now, the early human relationship with infection can be seen as one of gradual learning, shaped by observation, environment, and an enduring search for balance. 7. Nature's own antibacterial solutions. Long before humans noticed that certain substances could slow the spread of infection, nature itself had already been shaping microbial balance for millions of years. In the soil, in decaying leaves, on tree bark, and within waterlogged sediments, microorganisms have always lived side by side, sharing limited space and resources. In these crowded environments, survival does not depend on speed or strength, but on subtle chemical influence. Some organisms quietly release substances that limit the growth of their neighbors, not out of aggression, but as a means of maintaining space and balance. These naturally occurring antibacterial substances are the quiet ancestors of modern antibiotics. They were not designed with intent, but emerged gradually through evolution. Microbes that happened to produce compounds discouraging nearby competitors gained a small advantage. Over countless generations, these chemical abilities became refined and conserved, woven into the metabolism of certain bacteria and fungi. What humans would later call antibiotics were, in nature, simply tools for coexistence. Soil is one of the richest sources of these substances. Beneath the surface, countless microbial species compete for nutrients released from decomposing organic matter. This competition is not dramatic or violent. It is chemical. Bacteria and fungi release small molecules into their surroundings, subtly shaping which organisms can thrive nearby. Some of these molecules interfere with cell wall construction. Others slow protein production. Each compound nudges the environment in a slightly different direction. Fungi, in particular, play a notable role in this quiet chemistry. Living in damp, nutrient-rich environments, fungi often share space with bacteria. To maintain their niche, some fungi release compounds that inhibit bacterial growth. These substances diffuse slowly through the surrounding medium, creating invisible boundaries where bacterial expansion is less favorable. Over time, this chemical influence stabilizes the local ecosystem. Plants, too, participate in this process. Roots release compounds into the soil that influence microbial communities. Some of these compounds discourage harmful bacteria while supporting beneficial ones. Leaves and bark contain chemicals that slow microbial decay, protecting plant tissues while alive and shaping decomposition after death. Again, these effects are subtle and cumulative, rather than immediate. From a broader perspective, these natural antibacterial substances contribute to the overall balance of ecosystems. They prevent any single microbial population from dominating completely, encouraging diversity and stability. Without them, microbial growth might proceed unchecked, exhausting resources too quickly and disrupting larger ecological cycles. Nature's chemistry operates quietly, maintaining proportion rather than enforcing elimination. Human encounters with these substances occurred long before they were understood. Mold growing on food was sometimes associated with slower spoilage in surrounding areas. Certain soils were known to promote healing when applied to wounds. These observations hinted at underlying chemical processes, even if the mechanisms remained hidden. Over time, as scientific tools improved, humans began to isolate and study these natural compounds. Early researchers noticed that extracts from certain molds or soil samples could prevent bacterial growth in laboratory cultures. These findings were not dramatic revelations, but careful confirmations of what nature had been doing all along. Antibiotics did not emerge from nothing. They were discovered within systems that had already perfected their use. What makes these substances particularly gentle is their specificity. They do not sterilize environments. They do not remove all microbial life. Instead, they target certain bacterial features, influencing growth patterns without collapsing entire communities. This specificity reflects the evolutionary pressures that shaped them. An organism that eliminated all neighbors would eventually deprive itself of shared ecological benefits. When humans began refining these substances for medical use, they followed the same principle. Effective antibiotics interfere with bacterial processes that are essential yet distinct from those of human cells. This distinction mirrors the natural selectivity found in soil and plant chemistry. Modern medicine did not invent this idea. It is also important to recognize that natural antibiotics exist within a context of constant exposure. Microbes in the wild encounter low levels of many antibacterial compounds over long periods. This exposure encourages gradual adaptation rather than sudden collapse. Populations adjust, coexist, and persist. This slow dynamic is very different from the concentrated doses sometimes used in clinical settings, and it highlights the importance of respecting natural patterns when applying these substances. Nature's approach to microbial balance is patient. It relies on time, diffusion, and subtle pressure rather than force. Antibiotic compounds seep into environments slowly, influencing growth at the margins. This method allows ecosystems to remain resilient. When humans later concentrated these compounds, the effects became more pronounced, but the underlying mechanisms remained the same. Understanding antibiotics as natural substances reframes their role. They are not foreign chemicals imposed upon life, but part of life's own regulatory toolkit. Their power lies not in destruction, but in their ability to influence conditions under which bacteria grow. When used thoughtfully, they align with natural processes rather than opposing them. As this part settles, it may be helpful to imagine the soil beneath a forest floor. Leaves fall. Fungi spread delicate networks. Bacteria cluster and disperse, and chemical signals flow invisibly through damp earth. Within this quiet complexity, antibacterial substances are released gently, shaping relationships without fanfare. This is the environment from which antibiotics emerged. In the next part, attention will turn to the moment when humans first recognized and intentionally harnessed these natural substances, tracing the early discoveries that bridged nature's chemistry and modern medicine. For now, it is enough to rest in the understanding that antibiotics are not an invention of conflict, but a continuation of nature's long-standing practice of balance through subtle chemical conversation. 8. The discovery of antibiotics. The moment when humans first recognized antibiotics is often described as a discovery, yet it is more accurate to think of it as a gradual noticing. Nature had been producing antibacterial substances for millions of years, and humans had been benefiting from them unknowingly just as long. What changed was not the existence of these compounds, but human awareness of them. Through careful observation, patience, and quiet curiosity, people began to understand that certain substances could consistently slow or stop bacterial growth. In the early days of scientific medicine, infections were still mysterious in their origin, but patterns were becoming clearer. Researchers working with microscopes had begun to see bacteria, small living cells moving through water and bodily fluids. With this visibility came new questions. If bacteria were associated with illness, could something be found that selectively influenced their growth without harming the body itself? This question lingered for years, answered only partially by antiseptics that killed microbes broadly but were often too harsh for internal use. The transition from antiseptics to antibiotics required a shift in thinking. Antiseptics were designed to sterilize surfaces, eliminating as much microbial life as possible. Antibiotics, by contrast, would need to act with greater subtlety, interfering with bacteria while leaving human tissues intact. This was not an obvious goal at first, and progress toward it was slow and uneven. One of the most well-known moments in this story involves the observation of mold inhibiting bacterial growth. In laboratory settings where bacteria were grown in controlled environments, researchers occasionally noticed areas where bacterial colonies failed to thrive. Sometimes these clear zones appeared around accidental contaminants, such as molds drifting in from the air. Rather than discarding these cultures as spoiled, a few attentive observers paused to wonder why bacteria avoided these regions. In these quiet moments of curiosity, discovery took shape. It simply released substances into its surroundings that altered the local environment. Bacteria growing nearby found themselves unable to divide or maintain their cellular structures as effectively. Growth slowed, then stopped, leaving visible gaps in otherwise dense bacterial populations. Extracting and studying these substances required careful work. Researchers isolated compounds from molds and tested their effects repeatedly, observing consistent patterns. Certain bacteria were affected, while others were not. Human cells, when exposed under controlled conditions, remained largely unharmed. These early experiments revealed the possibility of selective antibacterial action, a concept that would soon reshape medicine. The development of antibiotics was not immediate after these observations. Identifying a substance was only the first step. It had to be purified, stabilized, and produced in sufficient quantities. It had to be tested for safety and effectiveness. Each step unfolded slowly, guided by trial, refinement, and repeated observation. There was no single moment of triumph, but rather a series of small confirmations that built confidence over time. As antibiotics became more refined, their effects in treating bacterial infections were remarkable. Yet even then, their success was understood best in context. They did not cure every illness. They did not work against viruses or non-bacterial conditions. Their power lay in their specificity, and with that specificity came limits. Early practitioners learned that antibiotics were tools, not universal solutions. The calm progression of antibiotic discovery also reflected broader changes in scientific culture. Collaboration increased. Findings were shared across institutions and borders. What one researcher observed in a laboratory could be tested and expanded upon by others elsewhere. This collective effort transformed isolated observations into reliable medical practices. Importantly, the discovery of antibiotics did not invalidate earlier medical knowledge. Practices like cleanliness, wound care, and supportive treatment remained essential. Antibiotics added a new layer of capability, but they did not replace the body's own healing processes. Instead, they complemented them, offering assistance when natural regulation alone was not enough. As antibiotics entered wider use, their natural origins remained evident. Many early antibiotics were derived directly from organisms found in soil or molds grown in controlled environments. Even as chemical modifications were introduced to improve stability or effectiveness, the foundational structures reflected their biological beginnings. This connection to nature reinforced the idea that antibiotics were not artificial intrusions, but carefully adapted natural compounds. The discovery phase also brought early awareness of caution. Researchers noticed that bacteria exposed to antibiotics over time could change in their responses. Some populations became less sensitive, continuing to grow despite the presence of substances that once inhibited them. These observations were not alarming at first, but they hinted at the adaptive capacity of bacterial life and the importance of thoughtful use. Throughout this period, antibiotics were seen not as weapons, but as quiet helpers. Their role was to create conditions under which balance could be restored. They slowed bacterial growth, reduced strain on the body, and allowed immune processes to function more effectively. This understanding shaped early medical philosophy around their use. The story of antibiotic discovery is therefore one of attentiveness rather than conquest. It highlights the value of noticing subtle patterns, of asking gentle questions, and of respecting the complexity of living systems. The substances that would transform medicine were already present, waiting not to be invented. As this part settles, it may help to picture a laboratory bench, a culture dish. In that quiet space lies the essence of discovery, not dramatic or forceful, but calm and revealing. The realization that nature itself held answers to bacterial illness did not arrive with fanfare, but with steady clarity. In the next part, attention will turn to defining more precisely what antibiotics are and how they are understood in simple terms. For now, the discovery of antibiotics can be appreciated as a patient unfolding of insight, guided by observation, curiosity, and a deepening respect for the invisible world that has always surrounded human life. 9. What an antibiotic is. In simple terms, after tracing the quiet path by which antibiotics were discovered, it becomes useful to pause and describe, in the simplest possible way, what an antibiotic actually is. Stripped of technical language and clinical associations, an antibiotic can be understood as a substance that gently interferes with the growth of certain bacteria. It does not bring instant change, nor does it act broadly on all forms of life. Instead, it works by influencing specific processes that bacteria rely on to live and reproduce. At its core, an antibiotic is selective. This selectivity is what distinguishes it from harsher substances that damage many kinds of cells at once. Antibiotics interact with features that are unique to bacterial cells or sufficiently different from those found in human cells. This allows them to slow or stop bacterial growth while leaving most human tissues unaffected. The effect is not dramatic or immediate, but gradual and measured. To understand this selectivity, it helps to recall the simplicity of bacterial cells. Bacteria lack the internal compartments found in human cells. Their genetic material floats freely, their ribosomes are smaller, and their cell walls are built from materials human cells do not use. Antibiotics take advantage of these differences. They are shaped, often at the molecular level, to fit into bacterial machinery in ways that disrupt normal function. An antibiotic does not usually kill bacteria outright. Many antibiotics are better described as growth inhibitors. They prevent bacteria from building new cell walls, from making essential proteins, or from copying their genetic material accurately. When these processes are slowed or interrupted, bacteria struggle to divide. Existing cells may persist for a time, but the population as a whole stops expanding. This pause is often enough for the body's immune system to regain control and restore balance. This cooperation between antibiotic and immune system is essential to understanding how antibiotics work. The medication does not act alone. It changes the conditions within the body, reducing the strain placed on natural defenses. Immune cells, no longer overwhelmed by rapidly growing bacterial populations, can clear affected areas more effectively. Recovery, in this view, is a shared process rather than a single decisive action. Antibiotics are also limited by design. Each antibiotic affects only certain types of bacteria. This limitation is not a flaw, but a reflection of their specificity. Bacteria vary widely in their structures and metabolic pathways. An antibiotic that interferes with one bacterial process may have little effect on another species that relies on a different mechanism. This specificity explains why antibiotics are not effective against viruses. Viruses are not cells. They do not have cell walls, ribosomes, or independent metabolic processes. They rely entirely on host cells to reproduce. Because antibiotics target bacterial features, they have nothing to interact with in viral infections. This distinction, though simple, is fundamental to responsible use. Another important aspect of antibiotics is that they are active only while present at sufficient levels. Once the substance is metabolized or excreted, its influence fades. Bacteria that remain may resume growth if conditions allow. This is why antibiotic treatment is typically prescribed for a set duration. The goal is not to maintain permanent suppression, but to support the body through a temporary imbalance. The simplicity of antibiotics as a concept can be misleading. While their purpose is straightforward, their effects ripple through complex systems. Bacteria do not exist in isolation. They form communities, interact with one another, and respond collectively to environmental changes. When an antibiotic is introduced, these communities adjust. Some populations decline, others fill available space, and overall balance shifts. In many cases, this adjustment is temporary. Beneficial bacteria recover over time, re-establishing familiar patterns. The body's internal environments are resilient, shaped by regular inputs and regulatory mechanisms. However, repeated or unnecessary use of antibiotics can alter these patterns more persistently, making balance harder to restore. This is why antibiotics are best understood as tools to be used thoughtfully rather than routinely. From a chemical perspective, antibiotics are small molecules, often derived from natural compounds. Their shapes and properties determine how they interact with bacterial components. Some bind tightly to enzymes, blocking their activity. Others integrate into cell walls, weakening structural integrity. Each interaction is precise, reflecting the long evolutionary history of these substances in natural ecosystems. Despite their precision, antibiotics are not perfect. They may affect beneficial bacteria along with harmful ones. They may cause temporary side effects as microbial communities adjust. These effects are usually manageable and reversible, but they remind us that antibiotics influence living systems rather than isolated targets. Understanding antibiotics in simple terms also involves recognizing what they are not. They are not cures in themselves. They do not replace the immune system. They do not strengthen the body directly. Instead, they alter conditions in a way that supports recovery. Their role is supportive and situational, not absolute. This perspective helps demystify antibiotics. They are not powerful forces acting independently, but carefully shaped influences that operate within the body's existing regulatory framework. When used appropriately, they align with natural healing processes. As this part settles, it may help to hold a gentle definition in mind. An antibiotic is a substance that selectively slows or stops the growth of certain bacteria, giving the body time and space to restore balance. It is neither aggressive nor passive, but quietly effective when conditions call for its use. In the next part, attention will turn to the specific ways antibiotics interact with bacterial cells at the cellular level, exploring these mechanisms with the same calm and unhurried approach. For now, the concept of an antibiotic can remain simple, grounded in selectivity, cooperation, and restraint, fitting naturally into the broader story of microbiology and balance. 10. How antibiotics target bacteria. With a calm understanding of what an antibiotic is, it becomes easier to explore how these substances interact with bacterial cells in practice. At this level, the process is not dramatic or forceful. Antibiotics do not attack bacteria in the way one organism might attack another. Instead, they influence specific cellular processes that bacteria depend on for growth and maintenance. These processes unfold quietly within each cell, guided by chemistry rather than intention. Bacterial cells, though simple, rely on a carefully coordinated set of activities to remain alive. They must maintain their shape, regulate what enters and leaves the cell, produce proteins, and copy their genetic material accurately. Each of these tasks depends on molecular structures and reactions that operate continuously. Antibiotics are designed to interfere with one or more of these tasks, gently interrupting the flow of normal cellular activity. One common target is the bacterial cell wall. As bacteria grow and divide, they must constantly build and remodel this wall to accommodate expansion and separation into new cells. The materials used to construct bacterial cell walls are unique, providing strength and rigidity while remaining flexible. Antibiotics that interfere with this process do so by blocking the enzymes responsible for assembling these wall components. Without properly formed walls, bacteria struggle to maintain their structure. This interference does not cause immediate collapse. Existing cells may persist for some time, but as they attempt to grow or divide, weaknesses appear. The cell becomes more vulnerable to internal pressure and environmental stress. Over time, growth slows significantly, and the population begins to stabilize or decline. Human cells, lacking these same wall structures, remain largely unaffected by this process. Another important target lies in protein production. Bacteria rely on ribosomes to assemble proteins from amino acids. These ribosomes are smaller and structurally different from those found in human cells. Some antibiotics bind specifically to bacterial ribosomes, altering their shape or blocking their function. When this happens, proteins, essential for metabolism and growth, are produced less efficiently or not at all. Again, the effect is gradual. The bacterial cell does not cease functioning instantly. Instead, it becomes less capable of maintaining normal activity. Enzymes are not replaced as they wear out. Structural proteins are not replenished. Over time, the cell's internal balance shifts, making growth and division increasingly difficult. Other antibiotics interfere with the copying of bacterial genetic material. Before a bacterium can divide, it must duplicate its DNA accurately. This process relies on enzymes that unwind the DNA strand, copy it, and reassemble it into stable forms. Certain antibiotics disrupt these enzymes, introducing errors or halting the process altogether. Without reliable DNA replication, division cannot proceed smoothly. Some antibiotics act on bacterial metabolism more broadly. They interfere with the production of essential molecules such as folic acid, which bacteria must synthesize on their own. Human cells obtain these molecules through diet, bypassing the need for internal production. By blocking bacterial synthesis pathways that humans do not use, these antibiotics achieve selectivity without direct harm to human tissues. What unites all of these mechanisms is their precision. Antibiotics do not introduce chaos into the cell. They create specific bottlenecks. They slow processes that bacteria cannot easily bypass. The cell continues to exist, but its ability to adapt and expand is constrained. This restraint aligns with the overall goal of restoring balance rather than eliminating life entirely. It is also important to understand that different antibiotics target different processes. Some focus on structure, others on metabolism, others on genetic replication. This diversity reflects the complexity of bacterial life, even in its simplicity. It also allows medicine to choose antibiotics based on the characteristics of the bacteria involved, tailoring intervention to context rather than applying a single approach universally. The timing of antibiotic action follows the rhythms of bacterial life. Antibiotics are most effective when bacteria are actively growing and dividing. During these periods, cell walls are being built, proteins are being synthesized, and DNA is being copied. When bacteria are dormant or inactive, these processes slow, and antibiotics have less immediate effect. Within the body, antibiotic molecules circulate through tissues and fluids, encountering bacteria wherever they reside. They do not seek out bacteria intentionally, but bind where conditions allow. Their distribution depends on factors such as blood flow, tissue permeability, and chemical compatibility. As antibiotics act, bacterial populations respond. Some cells fail to divide successfully, others persist temporarily without expanding. White blood cells can then clear affected areas more effectively, removing damaged cells and restoring tissue integrity. This cooperative process underscores an important point. Antibiotics are not replacements for immune function. They are assistants. Without immune responses, antibiotics alone are often insufficient to resolve infection. Conversely, immune responses may struggle when bacterial growth is rapid. Together, they form a balanced approach. At the same time, bacterial populations are not passive recipients of these influences. Over many generations, small variations may arise that alter how bacteria respond to antibiotics. Some cells may possess slight structural differences that reduce antibiotic binding. Others may produce enzymes that degrade antibiotic molecules. These adaptations emerge gradually, shaped by repeated exposure rather than sudden change. This adaptive capacity is not a sign of resistance in a dramatic sense, but a reflection of biological flexibility. Antibiotics introduce a new condition, and bacterial populations respond as they always have, through gradual variation and selection. This reality reinforces the importance of using antibiotics in ways that align with natural rhythms rather than overwhelming them. As this part settles, the image of antibiotic action can remain simple and calm. Picture a bacterial cell engaged in its routine tasks, building structures, making proteins, copying information. An antibiotic molecule enters this environment, fitting gently into a specific process, slowing it down. The cell continues for a while, then finds growth more difficult. Across many cells, this effect accumulates, shifting the population towards stillness rather than expansion. In the next part, attention will turn to why these interactions affect bacteria without causing similar disruption to human cells, exploring the biological differences that make selectivity possible. For now, the way antibiotics target bacteria can be understood as precise, measured, and cooperative, fitting naturally into the broader theme of balance that runs through the science of microbiology. 11. Why human cells are left unharmed. As the effects of antibiotics on bacterial cells become clearer, a natural question arises. Why do these substances interfere so effectively with bacteria while leaving human cells largely unharmed? The answer rests not in strength or resistance, but in difference. Human cells and bacterial cells, though both forms of life, are built according to distinct biological plans. Antibiotics work by recognizing and responding to these differences, interacting with structures that bacteria rely on but human cells do not possess. Human cells are more complex than bacterial cells. They contain a nucleus that encloses genetic material, along with numerous internal compartments that separate and organize cellular functions. These compartments allow processes such as energy production, protein modification, and waste handling to occur in specialized locations. Bacterial cells, by contrast, carry out all essential processes within a single shared space. This difference in organization creates opportunities for selective interaction. One of the most important distinctions lies in the presence of a cell wall. Most bacteria are surrounded by a rigid wall made from materials that provide strength and shape. Human cells do not have this structure. They are enclosed only by flexible membranes composed of lipids and proteins. Antibiotics that interfere with cell wall construction therefore have no equivalent target in human tissue. They pass by human cells without binding to anything essential. Another key difference involves ribosomes, the molecular machines responsible for building proteins. Both bacterial and human cells rely on ribosomes, but their structures are not identical. Bacterial ribosomes are smaller and arranged differently from those found in human cells. Antibiotics designed to bind to bacterial ribosomes fit precisely into bacterial structures, but do not interact effectively with human ribosomes. This structural mismatch allows protein production in bacteria to be slowed without disrupting the same process in human cells. Human cells also differ in how they obtain certain essential molecules. Some antibiotics interfere with metabolic pathways that bacteria must carry out internally. For example, many bacteria synthesize compounds such as folic acid as part of their normal metabolism. Human cells, however, acquire these compounds through diet rather than internal production. When antibiotics block bacterial synthesis pathways, human cells continue functioning normally because they are not dependent on those pathways. The way genetic material is handled provides another layer of selectivity. Bacterial DNA replication relies on enzymes that are distinct from those used in human cells. Antibiotics that interfere with bacterial DNA copying do so by binding to these specific enzymes. Human DNA replication, protected within the nucleus and governed by different molecular machinery. These differences did not arise to accommodate antibiotics. They are the result of separate evolutionary histories. Bacteria represent some of the earliest forms of life, while human cells evolved later, building upon and modifying basic cellular designs. Antibiotics simply take advantage of the divergence that already exists, fitting into bacterial systems while bypassing human ones. This selectivity, however, is not absolute. Human cells may experience indirect effects from antibiotic use, particularly through changes in microbial communities. Beneficial bacteria that normally support digestion or immune regulation may also be affected. These changes can lead to temporary discomfort or imbalance, even though human cells themselves are not directly harmed. Over time, these communities often recover, reestablishing familiar patterns. The body also plays an active role in protecting its own cells. Antibiotics are processed and distributed in ways that limit exposure to sensitive tissues. The liver modifies many antibiotic molecules, and the kidneys filter them from the bloodstream. This regulation helps ensure that antibiotics reach areas where bacteria reside while minimizing unnecessary contact elsewhere. It is also worth noting that antibiotics are typically used at doses carefully chosen to balance effectiveness and safety. These doses are sufficient to influence bacterial processes without overwhelming human systems. This balance reflects decades of study and refinement, informed by both biological understanding and clinical experience. The fact that antibiotics can be selective does not mean they are entirely benign. Like all substances that interact with living systems, they can have side effects. Allergic reactions, digestive disturbances, and interactions with other medications can occur. These effects arise not because antibiotics directly damage human cells. Understanding why human cells are left unharmed helps reinforce the idea that antibiotics are not blunt instruments. They are carefully shaped influences that operate within narrow biological boundaries. Their effectiveness depends on recognizing difference rather than applying force. As this part settles, it may help to imagine the body as a collection of diverse environments, each with its own structures and processes. Antibiotics move through these environments quietly, interacting where they fit and passing by where they do not. Their action is guided by compatibility rather than intention. In the next part, attention will turn to a related question: why antibiotics do not affect viruses, despite their effectiveness against bacteria. For now, the selective harmony between antibiotics and human cells can remain gently in mind, offering reassurance that these substances work not by harming the body, but by respecting the fundamental differences that define living systems. 12. Why antibiotics do not affect viruses. As the role of antibiotics becomes clearer, it is natural to consider their limits as well as their strengths. One of the most important of these limits is their inability to affect viruses. This distinction is not a shortcoming or oversight, but a direct reflection of the fundamental differences between bacteria and viruses. To understand why antibiotics do not work against viruses, it helps to step gently away from the idea of all microbes as similar threats and instead appreciate the quiet diversity of microscopic life. Viruses occupy a unique place in biology. Unlike bacteria, viruses are not complete living cells. They do not grow, divide, or carry out metabolism on their own. Instead, a virus is a small package of genetic material, either DNA or RNA, enclosed within a protective shell. Some viruses also carry an outer membrane, but even this structure is borrowed from the cells they infect. On their own, Viruses are inactive, drifting passively through environments until they encounter a suitable host cell. Because viruses lack cellular machinery, they do not perform the processes that antibiotics are designed to interrupt. There is no cell wall to build, no ribosomes to assemble proteins independently, and no metabolic pathways to disrupt. Antibiotics rely on the presence of these bacterial features. Without them, there is nothing for the antibiotic to bind to or interfere with. When a virus enters the body, it does not establish itself as a growing population of independent cells. Instead, it attaches to specific receptors on the surface of host cells and enters them. Once inside, the virus releases its genetic material and uses the host cell's machinery to produce new virus particles. Protein production, genetic replication, and assembly all occur using the host systems, not viral ones. This reliance on host cells is what makes viruses fundamentally different from bacteria. Any substance that disrupts viral replication by targeting these shared cellular processes would also harm the host cell itself. Antibiotics avoid this problem by targeting features unique to bacteria. Because viruses do not possess those features, antibiotics simply pass through viral infections without effect. The immune system responds to viruses in ways that differ from its response to bacteria. Specialized immune cells recognize infected host cells and work to limit viral spread. Fever, inflammation, and the production of antiviral signaling molecules all play roles in this response. Recovery from viral illness depends primarily on these immune processes rather than on external substances that directly inhibit the virus. Understanding this distinction helps explain why antibiotic use during viral infections offers no benefit. The antibiotic does not interact with the virus, nor does it speed immune recovery. Instead, it may influence bacterial communities that are not involved in the illness, altering balance unnecessarily. Historically, this distinction was not always clear. Fever, cough, fatigue, and pain could accompany both bacterial and viral infections. Without diagnostic tools, antibiotics were sometimes used broadly in the hope of benefit. Over time, patterns emerged that clarified their limitations. As scientific understanding improved, it became evident that antibiotics consistently failed to alter the course of viral illnesses. This observation reinforced the idea that viruses operate outside the scope of bacterial-targeted treatments. The development of antiviral medications followed a different path, focusing on specific stages of viral entry or replication rather than on general cellular features. The calm acceptance of this limitation is an important part of responsible antibiotic use. Antibiotics are not meant to address all forms of illness. Their value lies in their precision. When used in contexts where they are effective, they offer significant support. When used outside those contexts, they add complexity without benefit. This understanding also helps reshape expectations around illness and recovery. Viral infections often resolve through rest, hydration, and time, as the immune system gradually clears infected cells and restores balance. Antibiotics do not accelerate this process because they do not participate in it. Allowing the body to follow its natural course can be both effective and restorative. The difference between bacteria and viruses also highlights the diversity of life at the microscopic scale. Bacteria are independent living cells with their own internal processes. Viruses exist at the edge of what can be considered life, dependent entirely on other organisms to reproduce. Antibiotics are tailored to one of these forms, not the other. This specificity is a strength rather than a weakness. It reflects a deeper understanding of biological systems and an appreciation for difference rather than a desire for universal solutions. Medicine advances not by creating substances that act everywhere, but by developing tools that act where they are needed and nowhere else. As this part settles, it may help to hold a simple image in mind. Bacteria are small, self-contained workshops, busy with construction and production. Antibiotics enter these workshops and slow specific tasks. Viruses, by contrast, are instructions without workshops of their own. They enter existing spaces and redirect activity. Antibiotics, designed for workshops, have no effect on instructions alone. In the next part, attention will turn to differences among antibiotics themselves, exploring why some act broadly while others are more narrowly focused. For now, the distinction between bacteria and viruses can rest quietly, offering clarity and reassurance that limits in medicine are not failures, but reflections of the rich complexity of life itself. 13 broad-spectrum and narrow-spectrum medicines. As understanding of antibiotics deepened, it became clear that not all antibiotics act in the same way or with the same range. Some influence a wide variety of bacteria, while others are effective only against specific types. This difference gave rise to the idea of broad-spectrum and narrow-spectrum antibiotics, a distinction that reflects not hierarchy or strength, but precision and context. Broad-spectrum antibiotics are those that act on many kinds of bacteria. They are able to interfere with processes shared across different bacterial groups, affecting organisms that vary in shape, metabolism, and habitat. When introduced into the body, these antibiotics influence multiple bacterial populations at once. This wide reach can be helpful in situations where the specific cause of an infection is not yet known, allowing treatment to begin while further understanding develops. Narrow-spectrum antibiotics, by contrast, are more selective. They are designed to target particular bacterial groups or even specific species. Their molecular structures interact with features that are not widely shared across bacteria, limiting their effects to narrower populations. When the identity of the responsible bacteria is known, narrow-spectrum antibiotics allow intervention that is focused rather than expansive. This distinction is not about effectiveness in an absolute sense. Both types can be equally effective when used appropriately. The difference lies in how much of the microbial landscape they influence. Broad-spectrum antibiotics cast a wide net, while narrow-spectrum antibiotics place a careful hand on a specific thread within the system. The development of these categories emerged gradually as experience accumulated. Early antibiotics were often broad in their effects, simply because the first discovered substances interacted with common bacterial features. As knowledge expanded and diagnostic tools improved, it became possible to identify bacteria more precisely and to develop antibiotics that matched that precision. Using a broad-spectrum antibiotic can be compared to gently dimming the lights in an entire room to reduce activity, while using a narrow-spectrum antibiotic is more like turning down a single lamp. Both approaches can restore calm. Broad-spectrum antibiotics can be especially useful in urgent or uncertain situations. When infection is suspected but the specific bacteria involved are not yet identified, delaying treatment may allow imbalance to deepen. In these cases, a broad-spectrum antibiotic can slow bacterial growth across multiple possibilities, giving the body and clinicians time to respond more deliberately. However, this breadth also means that beneficial bacteria may be affected alongside harmful ones. The human microbiome, composed of many helpful bacterial communities, can shift temporarily under broad-spectrum influence. These shifts are often reversible, but they represent a larger adjustment than those caused by narrow-spectrum approaches. Narrow-spectrum antibiotics minimize this collateral influence. By targeting only the bacteria involved in the imbalance, they allow other microbial communities to remain largely undisturbed. This focused approach aligns closely with the natural principle of minimal intervention, influencing only what needs to be influenced. Choosing between these approaches involves balancing certainty and caution. When the cause of infection is clear, narrow-spectrum antibiotics offer precision and restraint. When uncertainty remains, broad-spectrum antibiotics provide coverage while understanding catches up. Neither approach is inherently better. Each has a place within thoughtful medical practice. This distinction also reflects a broader theme in microbiology: the value of specificity. Life at the microscopic scale is diverse and nuanced. Interventions that respect this diversity tend to preserve balance more effectively over time. Broad approaches may be necessary at times, but they are best used with awareness of their wider effects. From a biological perspective, the difference between broad and narrow action lies in molecular interaction. Broad-spectrum antibiotics bind to structures that are common across many bacteria, such as shared ribosomal features or widely used enzymes. Narrow-spectrum antibiotics interact with more specialized targets, present only in certain bacterial groups. These interactions are shaped by the evolutionary history of both bacteria and antibiotics. As bacteria diversified, so did their cellular features. Antibiotics derived from nature often evolved to influence specific competitors rather than entire microbial communities. Modern medicine, in refining these substances, has learned to preserve or enhance this selectivity when possible. The idea of spectrum also reinforces why antibiotics must be chosen carefully. Using a broad-spectrum antibiotic when a narrow-spectrum one would suffice introduces unnecessary change. Over time, such patterns can alter microbial communities more deeply, making future balance harder to maintain. Thoughtful selection helps preserve both effectiveness and ecological stability. It is important to note that the body itself often benefits from narrower influence. Beneficial bacteria contribute to digestion, immune signaling, and barrier protection. Preserving these communities supports overall health and resilience. Narrow-spectrum antibiotics, when appropriate, align with this goal by intervening only where imbalance exists. At the same time, the ability to use broad-spectrum antibiotics remains valuable. Life does not always present clear categories. Infections can involve multiple bacterial species or occur in contexts where rapid action is needed. Broad-spectrum tools provide flexibility, allowing medicine to respond to complexity without delay. As this part settles, it may help to view broad and narrow-spectrum antibiotics not as opposites, but as complementary tools. One offers reach, the other offers precision. Both are expressions of the same underlying principle, influencing bacterial growth in ways that support the restoration of balance. In the next part, attention will turn inward, exploring the human microbiome in greater depth and considering how antibiotics, whether broad or narrow, interact with this internal ecosystem. For now, the idea of spectrum can rest gently, illustrating how modern medicine continues to refine its approach by learning when to act widely and when to act with focused care. 14. The body's inner microbial community. As attention turns inward, the focus shifts from antibiotics themselves to the living landscape they influence within the human body. This landscape is known as the microbiome, a term used to describe the vast collection of microorganisms that live on and within us. Far from being an intrusion or anomaly, this inner microbial community is a natural and enduring feature of human biology. It develops early in life, adapts continuously, and remains present as a quiet companion throughout every stage of existence. From the moment of birth, microbes begin to settle on the skin, in the mouth, and along the digestive tract. These early arrivals are shaped by environment, contact, and nourishment. Over time, they form stable populations suited to the specific conditions of each region of the body. Warmth, moisture, available nutrients, and chemical signals all influence which organisms thrive. The result is not a uniform population, but a mosaic of communities, each adapted to its particular niche. The largest and most diverse of these communities resides in the digestive system, especially within the intestines. Here, trillions of bacterial cells live in close association with human tissues. They break down complex food components that human enzymes cannot process alone, releasing nutrients and energy that would otherwise remain inaccessible. In doing so, they contribute directly to digestion and metabolic balance. These bacteria also produce substances that influence the body in subtle ways. Some synthesize vitamins that support normal cellular function. Others generate short-chain fatty acids that nourish intestinal cells and help maintain the integrity of the gut lining. These interactions occur continuously and quietly, rarely drawing attention, yet they support processes essential to overall health. Beyond digestion, the microbiome plays a role in immune regulation. Immune cells lining the gut interact regularly with microbial populations, learning to distinguish between harmless residents and potential threats. This ongoing dialogue helps calibrate immune responses, reducing unnecessary inflammation while preserving the ability to respond effectively when needed. In this way, the microbiome acts as a training ground, shaping immune behavior over time. The skin microbiome performs a similar function at the body's surface. Bacteria adapted to the skin's slightly acidic environment occupy space and consume resources, making it more difficult for less compatible organisms to establish themselves. This quiet occupation acts as a form of protection, not through aggression. What is striking about the microbiome is its individuality. No two people share exactly the same microbial composition. Genetics, diet, environment, and life experiences all contribute to shaping these communities. Yet despite this variation, certain patterns remain consistent. Healthy microbiomes tend to be diverse, resilient, and stable, adjusting gradually rather than fluctuating wildly. This stability does not imply rigidity. The microbiome responds to change, just as bacteria do in any environment. Diet shifts, travel, illness, and medication can all influence microbial populations. Most of the time, these changes are temporary. Given supportive conditions, familiar patterns reemerge, reflecting the system's inherent resilience. Antibiotics interact with this system by altering bacterial growth conditions. When introduced, they influence not only bacteria associated with illness, but also some members of the microbiome. The extent of this influence depends on the spectrum of the antibiotic, the duration of use, and the individual's existing microbial balance. These changes are often noticeable only indirectly, perhaps through mild digestive discomfort or altered bowel habits. Importantly, the microbiome's response to antibiotics is not uniform. Some bacterial populations decline significantly, while others are less affected. This selective pressure can temporarily shift proportions within the community. Over time, as antibiotic influence fades, many populations recover, reoccupying familiar niches and restoring balance. This capacity for recovery underscores the adaptive nature of the microbiome. It is not a fragile system, easily broken by brief disturbance. Instead, it reflects millions of years of coevolution between humans and microbes. This long history has produced systems capable of absorbing change and returning to equilibrium when conditions stabilize. Understanding the microbiome also reshapes the way antibiotics are viewed. Rather than acting in isolation, antibiotics become one influence among many, shaping microbial ecology. Their effects are contextual, interacting with diet, immune function, and environmental exposure. This perspective encourages thoughtful use, aligning intervention with need rather than routine. The microbiome's role extends beyond physical health. Emerging research suggests connections between microbial communities and mood, behavior, and neurological function. While these relationships are complex and still being explored, they highlight the far-reaching influence of microbial life within the body. Again, these effects are subtle and cumulative, unfolding over time rather than appearing suddenly. What remains consistent across these observations is the theme of cooperation. The microbiome is not an adversary to be controlled, but a partner in maintaining health. Antibiotics, when used appropriately, support this partnership by addressing specific imbalances without undermining the system as a whole. This understanding invites a calmer relationship with both microbes and medicine. Rather than viewing bacteria as threats, They can be seen as participants in a shared environment. Rather than viewing antibiotics as blunt tools, they can be understood as precise influences applied with intention and restraint. As this part settles, it may help to imagine the body as a landscape with many quiet inhabitants. They move slowly, respond to conditions, and contribute to the stability of the whole. Antibiotics pass through this landscape like temporary weather, influencing growth patterns without redefining the terrain. In the next part, attention will turn to how antibiotics affect the microbiome in more detail, exploring the temporary shifts that can occur and the body's capacity to recover. For now, the presence of this inner microbial community can rest gently in mind, a reminder that health is not maintained by absence, but by balance, diversity, and ongoing quiet cooperation. 15. How antibiotics affect the microbiome. With an understanding of the body's inner microbial community, it becomes easier to consider how antibiotics interact with this living system. Antibiotics do not enter an empty space. They move through environments already inhabited by countless microorganisms, each adapted to its niche and participating in quiet, ongoing relationships. When antibiotics are introduced, they alter the conditions within these environments, influencing which microbes can grow easily and which slow down for a time. The effect of antibiotics on the microbiome is best understood as a temporary shift rather than a permanent disruption. Antibiotics are present for a limited duration. During that time, they apply selective pressure, reducing the growth of bacteria sensitive to their action. Some of these bacteria may be associated with illness, while others may be long-standing residents that contribute to everyday bodily functions. The antibiotic does not distinguish between intent or role. It responds only to cellular features. As sensitive bacteria decline, space and resources within the microbial community change. Other bacteria, less affected by the antibiotic, may occupy these spaces more readily. This redistribution is not chaotic, but adaptive. Microbial ecosystems are accustomed to change, responding continuously to variations in diet, environment, and immune signaling. Antibiotics represent one such variation, albeit a more pronounced one. In the digestive system, these shifts may become noticeable through subtle sensations. Changes in bowel habits, mild discomfort, or temporary bloating can reflect alterations in microbial activity. These experiences are usually short-lived, resolving as antibiotic influence wanes and familiar bacterial populations reestablish themselves. The gut environment, with its steady nutrient flow and immune interaction, supports recovery over time. The speed and extent of microbiome change depend on several factors. The spectrum of the antibiotic plays a role. Broad-spectrum antibiotics influence a wider range of bacteria, leading to more noticeable shifts. Narrow-spectrum antibiotics, by targeting specific groups, tend to leave much of the microbial community intact. Duration also matters. Short courses introduce brief changes, while longer exposure allows shifts to deepen before recovery begins. Individual variation is equally important. Each person's microbiome is unique, shaped by long-term habits and environmental exposure. This uniqueness influences how microbial communities respond to antibiotics and how quickly balance returns. Some individuals experience little noticeable change, while others may be more sensitive to temporary disruptions. Despite these variations, the microbiome's capacity for recovery is a consistent theme. Beneficial bacteria often persist at low levels, even during antibiotic treatment. Once conditions become favorable again, these populations expand, reclaiming their niches. This regrowth is supported by the body's regulatory systems and by the continued presence of nutrients and signals that favor familiar microbial relationships. Diet can influence this recovery process. Foods rich in diverse plant fibers provide substrates that support bacterial growth, encouraging the return of a balanced community. Fermented foods, which contain live microorganisms or supportive compounds, may also contribute to microbial resilience. These influences are gentle and cumulative, supporting recovery rather than forcing change. It is important to recognize that not all changes are negative. In some cases, antibiotics reduce populations that were contributing to imbalance, allowing healthier patterns to emerge. The microbiome is dynamic, and occasional shifts may lead to improved stability over time. What matters most is not the absence of change, but the ability to adapt and return to equilibrium. Concerns about antibiotics permanently harming the microbiome often overlook this resilience. While repeated or unnecessary antibiotic use can make recovery more challenging, a single, appropriately used course is unlikely to cause lasting harm. The microbial community has evolved alongside environmental fluctuations for millennia, developing strategies to endure and rebound. This perspective encourages a calm and balanced approach to antibiotic use. Antibiotics are neither harmless nor inherently damaging. They are tools that influence living systems, best used with intention and awareness. When used to address genuine bacterial imbalance, their benefits often outweigh temporary disruptions. The relationship between antibiotics and the microbiome also highlights the importance of timing. Using antibiotics only when needed allows microbial communities to spend most of their time in stable conditions. This stability supports diversity and resilience, making future disturbances easier to absorb. Thoughtful timing aligns medical intervention with biological rhythms rather than working against them. Understanding these interactions also reframes recovery. After antibiotic treatment, the body does not require aggressive measures to rebuild the microbiome. Given supportive conditions, recovery often occurs naturally. Rest, nourishment, and time allow microbial populations to adjust and stabilize. This gentle process reflects the broader theme of balance that runs through microbiology. It is also worth noting that the microbiome itself influences how antibiotics work. Bacterial communities can affect drug metabolism, absorption, and distribution. These interactions are subtle and complex, reminding us that antibiotics and microbes exist within interconnected systems rather than isolated pathways. As this part settles, it may help to imagine the microbiome as a garden. Antibiotics act like a temporary change in weather, perhaps a brief frost or drought. Some plants retreat, others endure, and when conditions return to normal, growth resumes. The garden does not disappear, it adapts. In the next part, attention will turn to side effects associated with antibiotic use, exploring how these experiences reflect shifts in balance rather than signs of failure. For now, the interaction between antibiotics and the microbiome can rest quietly, understood as a temporary and manageable adjustment within a resilient living system that has evolved to weather change with patience and grace. 16 side effects as signals, not failures. As antibiotics move through the body and interact with bacterial communities, their influence sometimes becomes noticeable in ways that are commonly referred to as side effects. These experiences are often framed as problems or complications. Yet within the calm perspective of microbiology, they can be understood differently. Side effects are not signs that something has gone wrong, but signals that the body and its microbial inhabitants are adjusting to a temporary change in conditions. When antibiotics are introduced, they alter the growth patterns of bacteria in multiple locations at once. The digestive system, rich in microbial life, is particularly sensitive to these changes. As certain bacterial populations slow or decline, the balance of metabolic activity shifts. Gas production, nutrient processing, and chemical signaling may change slightly, leading to sensations such as bloating, loose stools, or mild discomfort. These experiences reflect adaptation rather than damage. The body itself responds to these shifts in measured ways. Intestinal cells adjust their activity, immune cells recalibrate their signaling, and fluid balance shifts subtly. These adjustments are part of a continuous regulatory process that maintains internal stability. Antibiotics introduce a new variable into this system, and the body responds as it always does, through gradual recalibration. It is important to recognize that these sensations are usually temporary. The digestive system is particularly resilient, supported by constant nutrient flow and regenerative tissue. Given time and rest, balance reasserts itself without the need for forceful intervention. Some side effects arise not from microbial changes, but from the body's processing of the antibiotic itself. The liver and kidneys work to metabolize and eliminate these substances. In doing so, they may produce transient sensations such as fatigue or mild nausea. These experiences reflect the body's active engagement with the medication rather than harm to tissues. Allergic reactions represent a different category of response. These occur when the immune system identifies an antibiotic molecule as a threat and responds defensively. While less common, such reactions underscore the individuality of biological systems. What is tolerated quietly by one person may elicit a stronger response in another. This variability is a reminder that medicine operates within living systems, each with its own history and sensitivities. Viewing side effects as signals encourages attentive listening rather than alarm. Severe or persistent symptoms, by contrast, may signal the need for adjustment or reevaluation. In both cases, the body communicates through sensation, guiding thoughtful response. This perspective also helps counter the idea that effective treatment should be free of noticeable effects. Biological systems are dynamic. Change is often felt before it is resolved. Expecting complete stillness during adjustment overlooks the complexity of living processes. Antibiotics work not by bypassing the body's systems, but by engaging with them. Understanding side effects in this way fosters patience. The body is moving from one state of balance to another, guided by both internal regulation and external influence. This movement is rarely instantaneous. It is also worth noting that side effects vary widely depending on the antibiotic used. Different substances interact with microbial communities and metabolic pathways in distinct ways. Some antibiotics are more likely to affect digestion, while others may influence skin or nervous system sensitivity. These patterns reflect the diversity of antibiotic action rather than inconsistency in treatment. Supportive measures can ease this transition without interfering with recovery. Rest allows the body to direct energy toward regulation and repair. Hydration supports metabolic processes and elimination. Gentle nourishment provides substrates for both human and microbial cells. These simple actions align with the body's natural tendencies rather than imposing additional change. The concept of side effects as signals also reinforces the importance of completing prescribed courses of antibiotics when they are needed. Interrupting treatment prematurely may allow bacterial populations to resume growth before balance is fully restored. While discomfort may arise during treatment, it often resolves more smoothly when the process is allowed to unfold as intended. At the same time, this perspective encourages open communication and attentiveness. Not all side effects are minor, and not all signals should be ignored. Listening to the body and responding appropriately supports both safety and effectiveness. This balance between patience and responsiveness reflects the broader philosophy of microbiology as a science of observation and adjustment. As this part settles, it may help to imagine antibiotic treatment as a gentle tide moving through a coastal ecosystem. Water levels rise slightly, altering which organisms are most active at the shoreline. Some retreat, others adapt, and when the tide recedes, familiar patterns return. The movement itself is not a failure. In the next part, attention will turn to antibiotic resistance, exploring how bacterial populations adapt over time to repeated exposure. For now, side effects can be understood not as warnings of harm, but as temporary signs of adjustment within a living system that is responding thoughtfully to change. 17. Understanding antibiotic resistance slowly. As the discussion moves toward antibiotic resistance, It is helpful to approach the subject without urgency or alarm. Resistance is often described in dramatic terms, yet at its core it is a quiet, gradual biological process. It reflects the same principles that govern all living systems: variation, adaptation, and response to environmental conditions over time. When viewed calmly, antibiotic resistance becomes less a looming threat and more a natural outcome of interaction between organisms and their surroundings. Bacteria reproduce through repeated cycles of growth and division. With each cycle, genetic material is copied, and although this process is usually precise, small variations occasionally occur. These variations are not purposeful. They arise randomly as subtle differences in structure or function. Most have little effect on the bacterium's survival. A few, by chance, may influence how the bacterium responds to its environment. When antibiotics are introduced into this environment, they create a new condition. Bacteria sensitive to the antibiotic grow more slowly or fail to divide successfully. Bacteria that, by chance, possess traits allowing them to tolerate the antibiotic are less affected. Over time, these tolerant bacteria make up a larger proportion of the population, not because they are stronger or more aggressive, but because conditions favor their continued growth. This shift does not happen suddenly. It unfolds over many generations, often requiring repeated exposure to the same antibiotic. Each exposure gently nudges the population in a particular direction, favoring traits that allow persistence under those conditions. This process is slow and cumulative, reflecting the steady pace of biological change rather than abrupt transformation. It is important to note that resistance does not mean invincibility. Resistant bacteria are not immune to all influences. They remain subject to immune responses, environmental limitations, and other antibiotics with different mechanisms of action. Resistance simply means reduced sensitivity to a particular substance under particular conditions. Resistance traits can arise in several ways. Some bacteria may alter the structure of a target molecule, reducing the antibiotic's ability to bind. Others may produce enzymes that break down the antibiotic before it can act. Still, others may adjust how substances move in and out of the cell, limiting antibiotic access. Each of these changes reflects small modifications to existing systems rather than wholesale reinvention. Bacteria also have the ability to share genetic material with one another, sometimes even across species boundaries. This exchange allows resistance traits to spread gradually through microbial communities. Again, this is not a coordinated strategy, but a natural feature of bacterial life that promotes adaptability in changing environments. The emergence of resistance is closely linked to exposure patterns. When antibiotics are used frequently, especially when they are not needed, selective pressure increases. Bacteria encounter the same condition repeatedly, accelerating the gradual shift toward tolerance. Conversely, when antibiotics are used thoughtfully and sparingly, selective pressure remains low, and sensitive populations continue to thrive. Understanding resistance as a response to conditions rather than a failure of medicine reframes the conversation. Antibiotics do not stop working because they are flawed. Their effectiveness changes because living systems respond to persistent influence. This response is neither malicious nor avoidable. It is simply how life adapts. The body's own microbial communities play a role in this process. Within the microbiome, bacteria are exposed to low levels of antibiotics during treatment. Most recover once exposure ends. However, repeated or prolonged exposure may encourage the gradual accumulation of tolerance traits within these communities. Over time, this can influence how bacteria respond during future imbalances. This understanding reinforces the importance of completing prescribed antibiotic courses. Ending treatment early may leave behind bacteria that have been exposed but not fully suppressed, increasing the likelihood that tolerant populations persist. Allowing treatment to proceed as intended supports the restoration of balance more effectively. At the same time, It highlights the value of avoiding unnecessary antibiotic use. When antibiotics are introduced without clear benefit, they still apply selective pressure, influencing microbial populations without contributing to recovery. Reducing such exposure helps preserve antibiotic effectiveness over longer periods. It is also worth recognizing that resistance is not new. Long before modern medicine, bacteria encountered natural antibiotics in soil and water. Resistance traits evolved in response to these substances over millions of years. Modern antibiotics did not create resistance. They entered a world where adaptation was already an established process. What has changed is scale and concentration. Medical use introduces higher levels of antibiotics into specific environments, influencing populations more strongly than natural exposure alone. This amplifies processes that already existed, making thoughtful management essential. Scientific research continues to explore ways to slow the development of resistance. These efforts include developing antibiotics with new targets, refining diagnostic tools to improve precision, and studying alternative approaches that support microbial balance rather than applying broad pressure. Each of these strategies reflects an appreciation for biological complexity rather than an attempt to override it. Importantly, resistance does not signal the end of antibiotic usefulness. Many antibiotics remain highly effective when used appropriately. Resistance patterns vary by region, species, and context. This ongoing adjustment mirrors the adaptability of bacteria themselves, creating a dynamic equilibrium rather than a final outcome. As this part settles, it may help to picture resistance not as a wall being built, but as a landscape slowly changing under repeated weather patterns. Each exposure shapes the terrain slightly, favoring certain features over others. With care and restraint, the landscape remains varied and responsive rather than rigid. In the next part, attention will turn to thoughtful use and biological stewardship, exploring how human choices influence these gradual processes. For now, antibiotic resistance can be understood as a slow, natural response to persistent conditions, one that invites patience, understanding, and respect for the quiet adaptability of life at the microscopic scale. 18. Thoughtful use and biological respect. As understanding of antibiotic resistance deepens, the idea of thoughtful use comes naturally into focus. Thoughtful use is not a rule imposed from outside, but a practice that emerges from recognizing antibiotics as influences within living systems. Every time an antibiotic is introduced, it becomes part of an ongoing biological conversation, shaping conditions for microbes, human cells, and immune responses alike. Respecting this conversation means using antibiotics with intention, clarity, and restraint. Thoughtful use begins with acknowledging that antibiotics are not neutral substances. They are active participants in biological environments. When used appropriately, they help restore balance during periods of bacterial overgrowth. When used without clear need, they still exert influence, altering microbial communities without offering corresponding benefit. This understanding invites a shift away from habitual or precautionary use toward more deliberate decision-making. In many cases, the body is capable of resolving imbalance on its own. Immune systems have evolved over vast spans of time to manage microbial fluctuations. Fever, inflammation, and fatigue are not signs of failure, but tools the body uses to regulate internal conditions. Allowing these processes to unfold without unnecessary interference supports resilience and reduces reliance on external influence. When antibiotics are truly needed, thoughtful use also involves choosing the most appropriate type. This precision aligns intervention with the specific imbalance present rather than applying broad pressure. It reflects an ecological mindset, influencing only what requires adjustment. Duration matters as well. Antibiotics are prescribed for periods designed to support recovery without extending exposure beyond what is necessary. Completing a prescribed course allows the intended shift in bacterial growth to stabilize, reducing the likelihood that partially affected populations persist. At the same time, Avoiding unnecessarily prolonged treatment helps minimize broader microbial disruption. Thoughtful use extends beyond individual treatment decisions. It includes awareness of how antibiotics enter environments through agriculture, animal care, and waste streams. These pathways introduce low levels of antibiotics into soil and water, subtly shaping microbial communities outside the human body. Recognizing these broader influences encourages practices that limit unnecessary exposure at multiple levels. The concept of biological respect underlies all of these considerations. Respect does not imply avoidance or fear. It implies understanding and care. Antibiotics are powerful precisely because they engage with fundamental processes of life. Using them respectfully means acknowledging that power and applying it judiciously. This perspective also reframes the idea of stewardship. Antibiotics are not resources to be consumed, but tools to be preserved. Their effectiveness depends on maintaining conditions in which they remain useful. Thoughtful use today supports effectiveness tomorrow. Not through sacrifice, but through alignment with biological reality. Education plays a role in fostering this alignment. Understanding what antibiotics do and what they do not do helps set appropriate expectations. Antibiotics do not shorten viral illnesses. They do not prevent all complications. They do not replace rest, nourishment, and time. Recognizing these limits reduces pressure to use antibiotics in situations where they offer no benefit. Healthcare systems increasingly reflect this understanding through diagnostic precision and targeted prescribing. Advances in testing allow bacterial causes of illness to be identified more accurately, supporting narrow-spectrum treatment. These developments mirror the broader trend toward precision rather than breadth, reflecting respect for complexity. Individuals also participate in this process through attentive listening to their own bodies. Recognizing when rest and support are sufficient and when intervention is needed fosters a collaborative approach to health. Antibiotics become part of a broader toolkit rather than a default response. This approach does not deny the value of antibiotics. On the contrary, it preserves that value by ensuring antibiotics remain effective when they are truly needed. Thoughtful use enhances trust in medicine by aligning action with understanding rather than habit. At a deeper level, biological respect invites a change in perspective about humans' relationship with microbes. Rather than viewing bacteria solely as adversaries, they can be seen as co-inhabitants of shared environments. Illness represents a momentary imbalance, not a moral failure or invasion. Antibiotics assist in correcting that imbalance, not by erasing microbial life, but by guiding it gently back toward proportion. This perspective also encourages patience. Recovery is not always immediate. Biological systems respond over time, adjusting gradually. Trusting this process reduces the impulse to escalate intervention prematurely. Antibiotics work best when they complement natural rhythms rather than attempt to override them. As this part settles, it may help to imagine antibiotics as carefully placed stones in a flowing stream. They do not stop the water, but they influence its direction, slowing certain currents and allowing others to emerge. Used sparingly and thoughtfully, they guide flow without disrupting the stream itself. In the next part, attention will turn toward ongoing research and the future of antibiotic science, exploring how new approaches continue to reflect the principles of balance and respect. For now, thoughtful use can rest as a quiet guiding idea, reminding us that the most effective interventions are those that work with life rather than against it, honoring the subtle intelligence of biological systems shaped by time, diversity, and continual adaptation. 19. The ongoing study of new antibiotics. Even as antibiotics have become familiar tools in medicine, their study has never truly paused. The science surrounding them continues quietly, shaped by curiosity, patience, and respect for the complexity of microbial life. Rather than seeking dramatic breakthroughs, modern research often proceeds through careful refinement, observing how nature has long managed microbial balance and learning how to align medical practice with those principles. The search for new antibiotics frequently begins in the same places where the earliest ones were found. Soil, water, and living organisms remain rich sources of chemical diversity. Beneath forests, in ocean sediments, and within symbiotic relationships between plants and microbes, countless substances are produced that influence bacterial growth. Many of these compounds have existed for millions of years, quietly shaping microbial communities without drawing attention. Researchers approach these environments not as treasure hunters, but as listeners. They observe how organisms interact, which compounds appear when competition increases, and how balance is maintained over time. Advances in technology now allow scientists to identify and study these substances more precisely, isolating small molecules and understanding how they interact with bacterial cells at a molecular level. This work unfolds slowly. Most compounds discovered do not become medicines. Some are too unstable, others too weak, and some affect human cells as well as bacteria. Each candidate must be evaluated carefully. This process reflects the broader philosophy of antibiotic science: restraint, observation, and gradual progress. Alongside the search for entirely new antibiotics, researchers also revisit existing ones. By modifying molecular structures slightly, scientists can sometimes improve effectiveness, reduce side effects, or overcome specific resistance mechanisms. These adjustments do not reinvent antibiotics, but extend their usefulness, adapting them to changing biological conditions. Another area of study focuses on how antibiotics are delivered. The way a substance enters and moves through the body influences how effectively it reaches bacterial populations. Researchers explore formulations that release antibiotics slowly, concentrate them in specific tissues, or reduce exposure to non-target areas. These approaches aim to support precision and minimize unnecessary disruption. There is also growing interest in approaches that complement antibiotics rather than replacing them. Some studies explore substances that weaken bacteria without killing them directly, making them more susceptible to immune responses. Others examine ways to disrupt bacterial communication, reducing coordination within populations and limiting overgrowth. These strategies reflect a shift toward influencing behavior rather than enforcing elimination. Understanding the microbiome plays a central role in this ongoing research. Scientists increasingly consider how new antibiotics interact with beneficial microbial communities. Preserving diversity and resilience within these communities is now seen as an essential goal rather than a secondary concern. This perspective encourages the development of treatments that are effective yet gentle, aligned with the body's natural ecosystems. The study of antibiotics also extends into prevention. Vaccines reduce the need for antibiotics by preventing bacterial illness altogether. Improved hygiene, sanitation, and public health measures limit opportunities for imbalance to arise. Each of these factors reduces reliance on antibiotics, supporting their long-term effectiveness through reduced exposure. Education and communication are equally important areas of focus. Researchers and clinicians work to translate scientific understanding into practical guidance, helping individuals and communities make informed decisions. Clear explanations reduce misuse and build trust, aligning medical practice with biological reality. Globally, collaboration shapes the future of antibiotic science. Bacteria do not recognize borders, and neither does resistance. Sharing data, research findings, and best practices allows progress to unfold collectively rather than in isolation. This cooperative approach mirrors the interconnected nature of microbial life itself. Despite ongoing challenges, the tone of this research remains measured rather than urgent. Scientists recognize that antibiotics are part of a long story rather than a final chapter. Each generation contributes understanding, refining tools and approaches in response to observed outcomes. This continuity reflects the same slow adaptation seen in bacterial populations, creating a balanced dialogue rather than a race. Importantly, The future of antibiotic science is not defined solely by chemistry. It is shaped by philosophy. The shift toward precision, respect for microbial ecosystems, and alignment with natural processes marks a maturation of the field. Antibiotics are no longer viewed as blunt solutions, but as carefully placed influences within living systems. As this part settles, it may help to imagine a laboratory not as a place of urgency, but as a quiet space of observation. Samples are examined, patterns noted, and hypotheses tested gently over time. Progress comes not through force, but through understanding. In the final part, attention will turn toward reflection and closure, bringing together the themes of balance, cooperation, and quiet coexistence that run through the science of antibiotics. For now, the ongoing study of new antibiotics can be understood as a continuation of nature's own work, guided by patience, curiosity, and a deepening respect for the invisible world that sustains life. 20. Resting in a world of microscopic balance. As this long exploration of antibiotics and microbiology gently comes to a close, it is helpful to step back and allow the full picture to settle without effort. Throughout these parts, the focus has never been on urgency or conflict, but on balance, adaptation, and quiet cooperation. The microscopic world, though unseen, follows patterns that are steady and deeply familiar, mirroring the same principles that govern forests, oceans, and the slow rhythms of the human body itself. Bacteria are not strangers to us. They have always been present, shaping the environment long before humans appeared, and continuing to do so now. Within the body, they exist as residents rather than invaders, contributing to digestion, immune balance, and resilience in ways that are subtle yet essential. Illness arises not from their existence, but from moments when conditions shift and balance is temporarily lost. Antibiotics entered this story not as tools of domination, but as careful responses to those moments of imbalance. Drawn from nature's own chemistry, they reflect strategies that microbes themselves have used for millions of years to regulate their surroundings. When humans learned to recognize and refine these substances, they did not step outside nature's design, but learned to work within it. Throughout this journey, one theme has remained constant. Antibiotics do not act alone. They interact with immune systems, microbial communities, and the broader ecology of the body. Their effectiveness depends not on force, but on timing, specificity, and restraint. When used thoughtfully, they support the body's natural ability to restore equilibrium. The idea of resistance, often spoken of with concern, also fits quietly into this pattern. Resistance is not a sign that life has gone awry, but that life responds to conditions as it always has. Bacteria adapt gradually, generation by generation, reflecting the same evolutionary processes that shape all living systems. Human responsibility lies not in stopping this process, but in understanding it and acting with awareness. This understanding leads naturally to respect. Respect for antibiotics as powerful but limited tools. Respect for microbes as participants in shared ecosystems rather than adversaries. Respect for the body as a system capable of healing, given time, support, and appropriate assistance. When these forms of respect align, health becomes less about control and more about cooperation. Modern research continues to deepen this perspective. Scientists study not only how to create new antibiotics, but how to preserve balance, protect microbial diversity, and reduce unnecessary disruption. The future of antibiotic science is not defined by escalation, but by refinement, learning from nature rather than attempting to override it. For the listener, none of this requires memorization or vigilance. The purpose of this knowledge is not to create worry, but to offer calm clarity. Antibiotics exist because life is complex, not because it is fragile. They are used when needed, set aside when not, and always function within broader systems that tend toward balance. As this lecture ends, it may be helpful to imagine the body at rest. Breathing is steady, Digestion proceeds quietly. Immune cells continue their gentle monitoring. Microbial communities adjust and respond. The microscopic world does not rush, and neither must the mind. Understanding does not require tension. It emerges slowly, through exposure and repetition, much like the biological processes described here. The more one appreciates this pace, the easier it becomes to let the details soften and drift into the background. In this way, the science of antibiotics becomes not a technical subject, but a reflection on balance itself. Life is sustained through countless small adjustments, most of them unnoticed. When imbalance arises, correction follows through cooperation rather than force. Antibiotics are one expression of this principle, quiet and precise, doing their work and then fading away. As this final part settles, there is nothing left to solve or remember. The knowledge offered here is meant to accompany rest, not interrupt it. Like the microbial world it describes, it can remain present without demanding attention. The body knows how to return to balance. Life knows how to adapt. And understanding, once gently introduced, does not need to be held tightly. It can simply remain steady and calm as rest deepens and the unseen processes of life continue quietly on their own.